Yeah, I thought, yeah, but I hope you heard me. I'm MARCIS. I'm professor of medicine at the University of Latvia, also heading a research group that is called the Institute of Clinical and Preventive Medicine. And we do have three distinctive speakers for the session. So how we are going to organize, we are going first to have all the presentations. And since we do have only 3 presentations, not 4, likewise for the other sessions, we are expecting to have more time after the presentations for the discussions. So be ready, get your questions prepared, and I assume that we are going to have a lively discussion immediately after the presentations. So it's my great pleasure to call for the first presentation, Simon Pfisterer, on the topic of the metabolic disease and how really those issues are being approached in Finland. So we are really looking forward of your presentation.
So, can you hear me? Yeah. Great. Yeah, it's a really exciting opportunity to be here. Today has been an amazing day, and thank you for the invitation. Actually, I will talk about cardiovascular disease, not oncology, and I will not talk a lot about genetics. I will talk about the cellular consequences of the genetic aspects. So, how do all of these different genetic mutations actually affect a cellular activity, and how can we link that to disease mechanisms in action, and proposing a novel precision medicine application in dyslipidemia and cardiovascular disease. So I decided to give you a brief overview about cardiovascular disease. And to be short, it's the #1 cause of death. Every 40 seconds, a person experiences a heart attack. So within these, I thought it's 20 minutes. It will be 30 people who have a heart attack. Probably you have a family member or you know someone who experienced a heart attack or stroke. So it's a relevant issue. And the main contributing factor to this is high blood cholesterol, which is mainly transported in low-density lipoprotein cholesterol, LDL cholesterol. Probably you have heard of it as the bad cholesterol. But there are also other lipoprotein particles like VLDL, IDL cholesterol, which we know as remnant cholesterol, which is contributing to atherosclerosis and cardiovascular disease. So these particles can cross the blood vessel wall and they can form the formation of an atherosclerotic plaque, which leads to heart attack and stroke. And in a way, hypercholesterolemia is a global problem. In EU and US, more than 50% of the population have high blood cholesterol. So I think we really have to do something about that. So let's think about the precision medicine approaches in dyslipidemia. And I have to say, in a way, there is no real precision medicine application or approach to this. So what you can see here is the treatment guidelines, and I think it's pretty difficult for you to read, but in a way, you don't need to read this. In a way, how it starts is at the top. A doctor needs to make a decision, has a person a high blood cholesterol, are there other risk factors? And then the first decision is made, is this patient getting like cholesterol-lowering therapy? In nearly all of the cases, this is statin medication. Then there will be a follow-up after 6 months. The doctor will evaluate whether a patient has achieved blood cholesterol target levels or not. If this is not the case, they will think whether maybe they have to remind the patients taking the drugs, or they might consider giving an additional therapy, and so on and so forth. So the process goes in this regard. And what is also important in this, this is like based on population averages, knowing that low-intensity statin medication gives you 35 or 30% reduction in blood cholesterol, high-intensity 50%, on average. So this is what the doctors base their treatment decision on. But what we can see in studies like here by Ritger et al. In 2016, that individuals show very different responses to statins. So there can be some people who receive 70% cholesterol lowering. There can be some who don't achieve any cholesterol lowering effect. So in a way, this is more like, it's not a precision medicine approach, it's more like a trial and error approach in finding a right medication strategy. Going with statins, following up, escalating to ezetimibe, and maybe for some patients who have the, let's say, stamina to go through all of this process, maybe they get the medication that they really need. And what we can see, and a lot of studies actually show this, is that more than 80% of the high-risk patients actually never reach their cholesterol target levels. It takes a very long time to find an optimal treatment strategy for difficult patients. And you have to imagine this is happening in a situation where a person who already experienced a heart attack will have 20% chance of getting another heart attack in 2 years. So actually time matters. So my research group is kind of asking a slightly different question to this. So does biological variation contribute to treatment outcomes? And in a way, for this regard, we have to look at what are the biological processes underlying the development of dyslipidemia and drug action. And these are really cellular mechanisms, like there is LDL floating in the blood. It's taken up into the cells by a receptor called the LDL receptor and shuttled into the cells. And the cholesterol-lowering drugs actually affect that process via different mechanisms. Statins and PCSK9 inhibitors, these are drugs used in dyslipidemia, elevate the abundance of this receptor at the cell surface and thereby trigger more internalization into the cells. However, as I said, it's a cellular mechanism, so usually these are difficult to assess for individual patients. So therefore, my laboratory is combining cell biology, which provides insight into disease mechanisms, but which are highly manual and time-intensive, with automation, which is highly reproducible, high throughput, and low cost. And therefore, it's the goal to establish flexible, automated workflows in biological research. So, and we have set up an automated workflow which provides functional data at a single cell level. So we take a blood sample, we isolate white blood cells, and then we have robotic tools to look at how do these cells take up, for example, this LDL cholesterol, how do they store fat, and we have combined that with fully automated microscopy systems, automated image analysis systems, and data analysis systems. And therefore, we are able to get 26 cellular parameters for each subject. And if I would like to show them just a brief overview on how they look like, so we can really measure, like the top panel, you can see like really how cells take up, in this case, a fluorescent LDL particles. They get into certain regions within the cell, and we can really quantify a lot of different aspects for each individual cell. And we quantify for each patient thousands of cells. In the lower panel, you can see how we quantify lipid storage. So these yellow blobs are like your oil droplets in your salad sauce. It's like also in cells, they compartmentalize into these droplets. And we can quantify that. We can quantify each individual droplet within the cell. We can measure their size, and doing that for thousands of cells. And then it's getting a little bit more complex. So we do this under different treatment conditions. We do this not for only one cell type. We look at multiple cell types. We get lots of different parameters. We can look at the individual parameters, for example, only looking at how cells take up LDL, how they store fat. But then we can also integrate them into combination scores for uptake and storage, which we call lipid trafficking scores. And as a proof of concept, we have looked at patients with a genetic predisposition for high cholesterol. It's called familial hypercholesterolemia. They have a mutation, in this case, in the LDL receptor. And what is very interesting for us is that we looked at 21 individuals, 7 of them, for example, here in the center, have the same loss of function mutation. But what you can see here in the bars is that there is a lot of biological variation in the cellular outcomes for these individuals, even though they have the same monogenic mutation depicted in the scheme next to it. So we have a couple of individuals who have the same mutation, but then the cellular phenotype for them is very different. And what is even more exciting for us is that if we then look at those patients that are on statin medication, if we look at those patients who have poor cellular LDL uptake, for example, they have very high blood cholesterol values, whilst those that have very good cellular LDL uptake, they have very low blood cholesterol values. So in the end, those with a poor cellular readout have twofold higher blood cholesterol value whilst they are on statin treatment. So we can really pinpoint patients who would need more effective medication. So with this, I would like to summarize that part in a way. So we observe large inter-individual variation of cellular processes in these familial hypercholesterolemia patients, even if they have identical mutations. This cellular lipid uptake and storage associates with the blood cholesterol for these FH patients that are on treatment, and those with a poor cellular profile have twofold higher blood cholesterol values. So in a way, for us, the key question was, does this happen in the general population? And therefore, we went to a biobank study. It's the FINRIS 2012 cohort study. In 2012, they collected white blood cells, which we utilized. And for the same patients, we have access to laboratory values, genetic data, outcome data, NMR metabolomic data. We have data on drug reimbursement from 2002 to 2019 and causes of death. And we could show that the same thing is happening in the general population. So what you can see in this graph is that our cellular score is associating with blood cholesterol levels. And interestingly, the higher the intensity of the medication, the stronger is our correlation. So really showing that a person with a poor cellular profile will end up with a higher blood cholesterol concentration whilst they are on statin medication. And beforehand, I mentioned that cholesterol is not only transported in LDL particles, there are also additional particles that can lead to the formation of atherosclerotic plaques. And we can look into this with NMR metabolomics. And we did this for these patients. So also these intermediate density lipoprotein particles and small VLDL particles can cross the blood vessel and form atherosclerotic plaques. And what we can see that actually for those people that have a poor cellular score that are on treatment, we can see that there is a lot more cholesterol ester, more of those particles present. So in principle, these patients have really a pro-atherogenic lipoprotein profile whilst they are on medication. When we look at those patients and look whether they have actually achieved their treatment goal, if we don't do any stratification, we can see that 55% of the people who are on high-intensity statin reach their LDL target levels. However, if we then look at those patients who have a very good cellular score, all of them reach their target levels with statin medication. Whilst those that have a poor cellular score, more than 85% of them don't reach their target levels. So they would really need more effective combination therapy with additional drugs to get their blood cholesterol lowered efficiently. Beyond that, we can also see, even though it's a small group of patients, we can see that there is a profound impact in kind of myocardial infarction and stroke risk assessment. So when we look at how cells store fat, if people have problems in utilizing the stored fat, we can see that they have much higher likelihood of experiencing a heart attack. And if we combine even our LDL uptake and fat storage data, we can get even very high differences in how likely are these patients to achieve cardiovascular or to experience a cardiovascular event. And that's in a way super interesting, because these are people that are on standard lipid-lowering therapy. So we can actually pinpoint patients with very high unmet clinical need who we have to really identify and treat more effectively to prevent another event. And this is what we want to bring into a novel precision medicine application that combines the guidance for treatment optimization and together with an improved risk assessment for myocardial infarction and stroke. This will be for many different patient groups: familial hypercholesterolemia, in secondary prevention, where we could give actually the treating cardiologists the tools into their hands to really overcome problems in treatment of those patients. And of course, in primary prevention, really filtering out those people who would need already at early stage effective medication. And we want to move this into a spin-out company, so similar to Robert Greene this morning. So if you know some interested investors, angel investors, we are happy to take their numbers. So with this, I would like to thank the people in my group, the different funding sources, and of course also biobanks and computing infrastructure and business incubators and so on. And thanks to you for your attention. So questions later, or?
Yeah, questions are coming later. We are going to have the discussion. Thanks for the great presentation, and we are moving to the next one. The next one will be delivered by Vita Rovite, who is the most experienced person here in the room with the Latvian Genome Databank.
Thank you. So here I have my presentation. So thank you for this invitation to speak in this session. I will talk about one pilot project that we did based on our biobank, Latvian National Biobank, to identify hereditary cancer variants in early cancer cohort. And I will tell you about, okay, thank you. Firstly, a little bit about our biobank. We are Latvian National Biobank. Currently, we have participants of over 40,000 individuals from Latvian population. Our biobank is both population-based. Around 30% of the population of the biobank is population-based, and 70% of our biobank is recruited through different studies that are more or less disease-specific. Our biobank represents the whole population of Latvia, so whole regions of Latvia are represented and included in our biobank. And as a first line biological samples that we store, it is DNA plasma serum. We also store white blood cells. And for specific studies that we are doing, we are also storing gut microbiome tumor samples. And for example, we were also involved in COVID-19 research, so COVID related samples. We have possibility to link our biobank to national health records of Latvia. And we are doing extensive genotyping and omics data as far as we can, and constantly increasing different omics data on our biobank participants. And of course, we are open for collaboration with different partners on various studies, because on the graph, maybe you cannot see very clearly, but we have represented all different diagnosis groups from cardiovascular, oncology, endocrinology, so patients are involved in our biobank. The aim of this pilot study was to look specifically at the early cancer cases. I will go further into description how we define this cohort, but we wanted to select cases from our biobank, so donors that are already in our biobank that had cancer before 15 years old. And we also had this extensive questionnaire data where we ask about history in the family about different disease. And we wanted to specifically select these cancer patients with history of cancer in the family, so potentially potential carriers of some of inherited cancer variants, yes. We selected these patients. We wanted to do whole genome sequencing. Then we wanted further to study inherited cancer variants like in classical cancer genes like BRCA and other. And afterwards, we wanted to consult these patients, to identify these patients, consult them with consultation at clinical geneticists, and further potentially study new pathogenic variants that could be discovered in this cohort. So study's design was as follows. So we selected patients with cancer diagnosis from the age of 18 till 50. We didn't include childhood cases. We have specific project that's doing that in our biobank, looking at pediatric cancer. So we selected these cancer patients within specific age range, and we looked at family history, and potentially we prioritized cases where there were 3 cancer other affected relatives in the family, and so went down to the priority of less affected cancer cases in the family. We also looked at the death registry data that these people are still potentially don't have data that they have died in the death registry, because we wanted to contact them and get the information back to them if we discover something. And we have specific consent for our biobank where person can like check whether he wants to get some information back, and whether he wants or she wants to get some information back if there's something that could be done in the situation that is discovered. So in total, we were left with a little over 200 patients matching these criteria. And what we did whole genome sequencing, but not from all, but we utilized the resources that we had available, and we did we finally came up with 158 sequences of patients with whole genome. Further, I'm not a data specialist, but it was prepared by my colleague. We did standardized best practices GATAC analysis, and we specifically wanted to look at these inherited cancer genes, but also potentially to look for some novel variants. We also included some somatic variants using Cosmic cancer gene database. Afterwards, we get over 100,000 unique variants across all the samples. We removed common variants that potentially would not be causing this cancer, and we were left with 190 over thousands of variants. And afterwards, this is my colleague who did the all analysis, right, specialist. I really acknowledge him in this project. Afterwards, we looked at available prediction tools and databases to look really for these pathological, potentially pathological variants that have been described or are likely pathogenic for causal variants of cancer. And also looking at these international guidelines. Afterwards, the numbers are very small in the picture, but we came up with over over 200 relevant variants that were still further reviewed with clinical geneticists who went through whole variants. And our clinical geneticist involved in the study, I could not find a picture of her, but also great thanks to her. So she reviewed this, and at the first step, it was in this year, May, we came up with the first list. It was like first list preliminary results with 33 patients. We had these potentially clinically relevant variants we wanted to report back to these participants. So far from the May, we have some break in summer. We have consultations with 20 patients that we have identified. And for all of these, we have done independent genetic test for confirmation of this mutation in independent diagnostic laboratory. So for 8 of these patients, we actually confirmed these mutations, and these families are currently under high-risk screening. I will show 2 of the families further in my presentation. For 3, it was not confirmed, but this is a bias that could happen in such studies using NGS, but of course, we have also internal discussions that, of course, these patients has other variants that we just filtered out during screening, and maybe we need to look at them more thoroughly, and for one is still under analysis. So one of the families that we discovered was this family of familial adenomatous polyps syndrome in mutation in APC gene. The patients with this condition develop lots of polyps in colon and rectum, and there are around 100% probabilities that they will develop colorectal cancer in their lives. So you can see this family with all these potentially affected members, and according to NCCN guidelines, they are currently appointed with colectomy, and the patients are very adherent to that because they understand the risks. And as you can see, there are many other family members that are affected by this condition, and potentially this could improve the further life of this family. The second family was an interesting case where a young woman, 33 years old, had breast cancer history, and after sequencing, we were surprised that we discovered this Lynch syndrome related variants. Lynch syndrome is inherited cancer condition where cancer are more likely to develop in different sites of gastrointestinal tract, and actually in the history family of this woman, we found that there were several cases already with different conditions in gastrointestinal tract, and of course the whole family further are undergoing this screening and consultation with clinical geneticists, and actually this case is also under review at Molecular Tumor Board of Riga East University Hospital, potentially because of course there are these very clear recommendations for what you need to do for Lynch syndrome surveillance, but whether there are specific things you need to do also for surveillance or screening of breast cancer in this case, because patient had the previous history of breast cancer. So these were the 2 main families I wanted to show at this point. But currently in this project, we are in the situation as illustrated in the graph. We only have some preliminary results undertaken and looked at, but underneath there is still so much data and so much questions we want to ask. And as from these 8 families, we already have seen that we can improve significantly improve potential health trajectories of these patients. So overall, we have demonstrated that this is feasible approach for identifying such cases. We have 8 families identified and so far, but we are going to study our data further. We have also discovered new potentially pathogenic variants that we will investigate. And we have several also discussable questions that we would like to address further and will do within this project. One is this breast cancer case was not the only one where we have this mutation in, for example, Lynch syndrome genes, but the patient has breast cancer. And we have some other cases as well where the actual mutation or gene where we have found this mutation does not correspond to cancer types the patient has, but he or she has this history of. So potentially that is the thing we want to look further and maybe also look deeper. Maybe there is additional variants that these patients are carrying that are contributing to specific types that we just could not do at the first step. And the second is really understand and evaluate how we proceed with pathogenic variants that are in known cancer genes. Potentially they are predicted pathogenic, but there are no reports in literature that they are related. So potentially maybe they are new and potentially maybe they are pathogenic and affecting some families. But we need to look deeper and still work on the data, so we have a lot of work to do still further. And with this, I want to finish description of my pilot study, and I want to acknowledge my colleagues, right, specialists in BioLabs who were very directly involved in this project. I would like to acknowledge personnel at our National Biobank and Genome Center, and also I want to thank 2 professors who conceptually contributed and supported this project conceptually, Professor Klovinch and Professor LIAA. So thank you very much.
Thank you indeed. And now it's my great pleasure, and we are really looking forward to hear the data from United Arab Emirates on the Genome Project, including 500,000 individuals. And the speaker, it's my great pleasure to introduce to Dr. Ahmad Al-Awadhi. So the floor is yours.
Thank you, Mr. President. Good afternoon, Your Excellency. Good afternoon, distinguished guests. I'm pleased and honored to be with you this afternoon in this hall. And I would like to thank the organizers. I would like to thank everyone who contributed in the success of this forum. Before I introduce myself, I do have a secret. I couldn't hide it. I have to reveal it in front of all of you. Usually people fall in love from first glance, but myself and my colleague Khalid, the moment we stepped out of the Riga airport, we fall in love with the city. So, okay. And I'd like to thank the organizers for joining me at this Precision Medicine Network Forum. My name is Dr. Ahmad Al-Awadhi. I'm Director of Community Outreach at M42 and the Manager of the Emirati Genome Program. And I'm going not to talk about diseases, not to talk about genetics. I will talk about an exciting program, an exciting talk to give to you that I'm passionate about. I will be introducing you to the groundbreaking initiative that holds immense promise of the future of healthcare in the United Arab Emirates, the Emirati Genome Program. The reason that I'm presenting this presentation about the Emirati Genome Program, because in very short time in the United Arab Emirates, we have already collected more and sequenced more than 500,000 samples. And this is, I believe it's a record, again, less than 3 years. But before embark on the genomic journey, let me provide a brief overview of M42. Earlier this year, through the integration of G42 Healthcare and Mubadala Health, M42 was formed, a first of its kind unified healthcare powerhouse by harnessing innovative solutions, unique medical and data-centric technologies, including genomics and AI. M42 is transforming the traditional healthcare ecosystem and delivering the highest level of precise, patient-centric, and preventative care. Now, for a crowning example of what M42 can deliver, the Emirati Genome Program. This initiative stands as one of the most ambitious and comprehensive population genomic projects ever taken. In terms of the size of the sample, we are targeting million. And in terms of the technology that we are using, we are using the latest technology from third industrial revolutions, supported by second and third generation, and that means the latest technology of those giant machines for sequencing the genome. Its aim to explore and generate genetic makeup of every Emirati citizen. This resulting in reference genome will lead a better preventative health care for the nation of the United Arab Emirates, utilizing the latest in whole genome sequencing and artificial intelligence technologies. EGP aims to create the highest quality and most comprehensive genome data, unlocking a new biological insight. There is no doubt that the Emirati Genome Program is paving the way of a new era of healthcare, a future where medicine, personalized, preventative, and precise. The program targets widespread diseases such as obesity, diabetes, hypertension, cancer, asthma, and other diseases. It's a testament to our commitment to advancing healthcare through precision and personalized medicine. How does it work? The program invites Emirati citizens voluntarily take part in the study by visiting one of the sample collection sites and providing a single blood sample. Then, with the help of advanced sequencing technology and artificial intelligence, we are able to generate and analyze comprehensive genomic data and produce reference genome specific for the UAE citizens. From the moment of sample is collected, it becomes part of the transformative process. Our commitment to data quality and patient privacy is unwavering, ensuring that each sample contributes meaningfully to the greater understanding of genomic, with the sample safety and ethical use never compromised. We have come along way since G42 Healthcare appointed to manage the Emirati Genome Program in 2019. The G42 Healthcare appointed to manage the Emirati Genome Program in collaboration with Department of Health in Abu Dhabi. In November 2020, first collected site opens at Abu Dhabi National Exhibition Center, ADNIC. And we welcomed the participant. On January 2021, launch of Omics Center Lab, Omics Center of Excellence, the region's largest and most advanced Omics Center to facilitate Emirati Genome Program. And this laboratory, the Omics Center of Excellence, one of the largest, if not the largest, laboratories in the region. On January 2021, introduced of home collection. We do home visits. Our families back home extend the members of the family numerous. So, and instead of asking them to come to our fixed center to donate and participate in the Emirati Genome Program, we do have home visit. Let me tell you this story. Once we went to this family and we found the member of the family, 80 persons. So we make their life easy. So we went to them, we collect. After they called and they said, we want to participate in the Emirati Genome Program. On January 2021, more than 20 healthcare providers partner with the Emirati Genome Program to welcome participants. On September 2021, we hit 100,000 samples. On March 2022, more than 200,000 participants contribute to the program as 60 plus government entities helped encourage participants. On March 2023, we reached to 400,000 samples. And bear in mind, collect samples and sequencing at the same time. On January 2023, more than 100 locations are live across the United Arab Emirates. Welcome the participants. September 2023, we have already hit 500,000, more than 500,000 samples, and we sequence them. And this is amazing job. I would like to thank everyone who contribute and participate and work in this program. In conclusion, the Emirati Genome Program is not just a scientific endeavor. It is a commitment to the well-being of our community, facilitated by G42 Healthcare and now M42. Its aim to analyze the complete genome of the United Arab Emirates citizen and allowing researchers, doctors whom it may concern in this field to identify the causes of genetic diseases, study in deep the mutations, number of the mutations, strength of mutations, locations of those mutations, the chromosomes, to develop effective treatment and prevention plans. Before I finish my talk, again, thank you for your attending and time. Again, thank you for the organizers. Thank you for inviting me for this speech. And Thank you for your attention. Thank you very much.
And I would ask you to stay here on the stage, and also the other speakers come up to the stage, and the floor now is open for questions, for discussions. So please be active, and I think those have been 3 very interesting presentations from different parts of the world and with different potential of prevention. So please. Meanwhile, actually, what I wanted to ask to the last presenter on the Arab Emirate program, what metadata are you collecting? Are you linking those individual genomic data also to the laboratory data, to the data describing maybe certain diseases, metabolic syndromes, or cancer data? Do you have this linkage to the diagnosis as well from the clinical part?
Yes, it will. Yes, it will soon. We do have Malafi, and we do have this centric data, and now we are doing a lot of researches. Also, we are working on the clinical genome for the patients. Also, we are working in premarital tests. So the data is owned by Department of Health, and we have saved this data in the national clouds. So soon will be revealed for the patient as long as we finish. In this program, the Emirati Genome Program, we are attending to draw the genetic map for the UAE citizens. And after, as I just mentioned, to give the researchers, scientists, doctors, what may concern this field, to study those causes of the genetic diseases, causes of the mutations, and planning to write down the prevention and medical plans. And they cannot do that unless they see the data, unless they see what type of mutations do we have.
Thank you. And to continue with this, so you are allowed really to use those research data for clinical applications, yes? So those are clinically certified laboratories that are doing this, and it is no problem for using this for clinics, right?
Yes, yes. Mm-hmm. We use this for nowadays, we use it for clinical genome.
Yeah, thank you. Thank you. That's really very exciting. Any other questions? My question to Vita actually might be whether did you find among those individuals also CDH1 gene mutations that could be characteristic for hereditary gastric cancer, but also some other cancers?
We currently, I cannot answer this question because I don't know by heart. We haven't checked specifically, but we can do that. Overall, there were some also patients with gastric cancer included overall in the cohort that I know. So we can check that, yes.
Thank you. And actually, I do not see any other questions from the floor, but please once more come to the front and ask questions. And another question maybe to Simon on this. This is exciting technology, and how you really plan to introduce it to the real practice. So you mentioned also the family hypercholesterolemia, but as I understand, it is still more not in familial cases, not in genetically determined cases, but this could be really applicable to routine clinical application. Yeah.
Yeah, I mean, great question. Thank you. I mean, I think there are applicabilities for all of them. So I think what has been really exciting for us to see that there are patients that have identical, in familial hypercholesterolemia, genetic testing is often used to confirm that they are familial hypercholesterolemia. But still, I mean, LDL receptor represents 90% of all of the FH mutations, we know 3,000 mutations in LDL receptor, but for less than 10% of them, we know whether they have a consequence. Also, what Robert Green showed this morning, that he only focused on pathogenic and likely pathogenic variants. So, but we know that there are 4-fold more variants of unknown significance. So I think that's a very interesting question. So in principle, there are a lot more people walking around with these problems, and I think the technology that we are using can really address that. And it can provide more in-depth information, even if a clinician has a genetic test for a patient, because we can see that even if patients have the identical mutation, there is still a lot of biological variation that can lead to different treatment outcomes and disease outcomes. And I think that's linked in a way that I think genetics is great, but there's also great lack of information still. There's a lot that we don't understand. And I think when we look at the cellular readout, we just look at the combined effects of all of this genetic lifestyle, and that gives us a very good readout for looking at the treatment outcomes for that. And therefore, it's also working for the general population, where it's more like a combination. And I think we all walk around with some pathogenic, some likely pathogenic, other variants, and it's all a mix of that. And that's what we can read out also in the general population. So in the general population, we have people that have a similar cellular profile to a person with severe familial hypercholesterolemia, and this person has no monogenic identified mutation. Even if you have clinical diagnosis of FH, only 20% of them you find a monogenic, pathogenic mutation. So I think there is great application for both scenarios, and especially secondary prevention of heart attack, where people have 20% of chance of getting another heart attack, and you really need to act fast to get blood cholesterol levels down, and it's not happening at the moment.
Thank you. Actually, there are our colleagues that are running also the family hypercholesterolemia registry here in Latvia. Unfortunately, they are not presenting on this meeting, maybe during the next year. But another fast question before we are opening once more to the floor to you. Are those results, the lipid particles, as I understand, are they changing over the time, or are they specific for a particular individual in dynamics?
Okay. I'm not 100% sure which way you want to go with the question, but I mean, let's say, I think for each individual we have a certain, the cellular profile will affect how fast you can internalize your LDL particles, and that determines how long they are staying in the circulation. If they stay longer in the circulation, you have a propensity that they are getting more atherosclerotic, or prone to induce atherosclerotic plaques, and that's what we can see. So I think each one of us has a little bit different turnover of lipoprotein particles. However, the lipoprotein particles that we use for our assay are in a way standardized, because we want to be able to compare the results for a lot of different people, and also over time, like 2 years ago now, can we compare the result from maybe you or maybe you?
Yeah, that was just my idea, whether you are giving for serial tests depending on the therapies. But okay, the microphone is open here. Yes.
Thank you for your presentations. I have a question for Dr. Simon. So it's clear that low density cholesterol, it's associated with heart attack, but the high levels of it. But what about the other side? What about the too low levels of this specific low density lipoprotein cholesterol? Are there any health risks associated with it, or are there any? Because when I read up on it, there were no clear consensus in literature if there are any health risks associated with it. Yep.
Yeah, I would say, I mean, I'm not a medical doctor, I'm a biochemist, so, but the overall consensus is at the moment, the lower the better. And in a way, we are all, even what we consider as normal is probably too high. Like a LDL cholesterol concentration of 2.5 to 3.5, it's still such that, and that's kind of an interesting question for these people. There might be some people who are more prone to get cardiovascular disease, even though they're in this normal range, for example. Then if you go lower, and I think there it's very interesting, if you look even at the literature of like, which is 40 years old, from Brown and Goldstein, who got the Nobel Prize for taking FH patients in this case and deciphering clathrin-mediated endocytosis, how do LDL particles get into the cells? And they have said that in all the animals that are walking around and in young, very newborns, average LDL cholesterol, now it's kind of 50 milligrams per LDL, and what the average that I just said is something like 190, 150 milligrams. So it's like 3 times, 5 times lower. And they have been suggesting that like this very low level would be actually beneficial for us. But of course, as our lifestyle has like completely changed and we are not living like animals anymore, we are, we are like in a higher level, which is of course like from a long-term perspective, detrimental to our health, but nature doesn't care about that. It's all about whether we can reproduce. So I think from this perspective, you know, it's a difficult question, but I think there are studies that really show that bringing blood cholesterol down to these low levels that Brown and Goldstein mentioned, there are no severe side effects. But then, of course, you need also some lipoprotein particles in your blood. I mean, nature would have not evolved such a complicated system if you could just eradicate it completely. We needed to transport nutrients within our body. We needed to transport vitamins as well. So you can't kind of deplete it completely, but I think you can go to a very, very low level with that. But you need to maintain kind of the core functionality of it, but then, yeah.
Okay, thank you.
Yeah, other questions?
Hi, everyone. Dr. Ahmad, many thanks for your presentation. It was wonderful. During several years, as I understand correctly, you received 500,000 of population in your biobank. But the question is that what I want to ask is more technical, because in Europe, there are several problems with the secondary data use. Because must be a platform that must use the data, the metadata, etc. How do you tackle this problem? Is there any regulation that helps you consent, etc.? So how do you solve the issue of the secondary data sharing? Thank you.
Thank you very much. First of all, I would like to say the Emirati Genome Program is hope probe, and the main aim is to create healthier Emirati population and healthier Emirati society. Yes, we do have consenting. The participant is allowed from all ages, from newly born until whatever ages. Also, we consent below 18 years. Parents should sign the consent form. This is for the Emirati Genome Program. And if there is some clinical genome, again, there is another consent form. If below 18 years, the parents should and must sign. If it's above, it's adult, he can sign by him or herself. There's a regulation. We are working under the umbrella of Ministry of Health and Prevention. Whatever regulation they have, we follow. We cannot do things from our own. Mm-hmm. See, in the United Arab Emirates, 3 regulatory three umbrellas that we work under them: Department of Health in Abu Dhabi, Department Health Authority in Dubai, and Ministry of Health and Prevention. So we work under their umbrella and their regulations.
Mhm. Yeah, thank you.
Thank you.
Maybe just one thing that I also want to mention. So the question is, there is a possibility to use the secondary data use. You can your participants can give and share data to industry, to other researchers who want.
Yes, in the consent form it says that we can use this data and researches, but without revealing the personal data, personal identifications. Now we are working with Longenesis. We use their software, and also, as I mentioned in the presentation, we save. This is the lights. Okay, we save the data and the national clouds, and nobody can look at these personal data because it's protected under the law of the protection of personal data of the United Arab Emirates. But we can use the results and research or in any conference or. I hope I answered your question.
Yes, thank you. Yeah,
Okay.
thank you.
Again, your personal data is protected. Nobody can look because the moment the participant comes to participate, the moment he put his ID in the reader, all his personal information will be encrypted, taken with a barcode. So there is no personal data shown, only barcode.
So it means only metadata, yes?
Yes.
Okay, thank you.
Thank you.
Thank you. But just to continue with the same question, so for the medical purpose, you are using the personalized data. You are not doing any pseudonymization for, definitely yes, for further research. But just since you are implementing this in clinics, you are still working with the personalized data, right?
Yes, but they have to consent again.
Absolutely. Okay, I see. And maybe could Vita comment how it's done in Latvia? And don't we have problems with really because the state genome data registry doesn't have the personalized data, the keys lying elsewhere?
Yes, we have quite a complicated system actually also for our study to get back to the participant because our current genome research law was developed in early 2000s when it was not the research field was not so clear yet how it will progress and we have strict division from this personal data that is in genome registry, and in the biobank we also have this coded data that we work with, we can research, but to get back to participant we need to have specific procedure where we go through these genome registries that contact the person back. So in a sense it's very good because the person is protected, but if you really have this clinical need or clinical use of some research data that could really impact the life further or health of the patient, of course it would be very good to have more flexible system with specific consents as in your case to really get back to the participant. Yes, so I hope that we could change our legislation so it would be more accessible in Latvia and more doable as well.
That was my question. What we should do to make it more really functional and better use for clinical purpose?
Of course, as you, Professor, know, but maybe not all here in the room, that we have developed new biobanking law in Latvia that would allow us more flexibility, but maintaining this also respect and the protection of the patient's data. So I believe there is a clear case that we need to change our legislation to be able to more impact, to generate more impact on the lives of.
Yes.
In this case, they can use the PI specific person or the who owns the data can have this PI personal. He can look at if somebody asks for clinical genome, for example, he will make a request. We need this clinical genome and these genes to be tested for this and this. The only one person or two person who owns the data can take the personal data from the barcode and then attached with the results.
But still, there are researchers or persons that do have the access to the data. And
No. It's if
Yeah.
nobody, even the one he's authorized to look at the data, if he looked with no reason,
Upon the
would be in.
request. Upon the request,
Yeah.
yes. And I think we can come back a minute later to the really biobanking law. But meanwhile, let's take a question from the audience.
Thank you for a wonderful presentation. I have a question to Professor Pfeiffer. My question would be, did you observe the age difference in the poor absorbers of cholesterol? And does it mean that they have more aggressive atherosclerotic disease at a younger age?
Yes, we observed a small correlation that like cellular LDL uptake is decreasing with age. But it's only a small one. And I think that also comes along with, I think that can underlie why people are developing like higher blood cholesterol levels with age. Did it answer your question? Yes.
The question was more, if the person is having this low absorption levels, does it mean that he has higher risk for aggressive cardiovascular disease further on, let's say at the age of 40, more aggressive?
I think, of course, like if they have a very poor cellular readout, poor LDL uptake, poor lipid storage phenotype, then they are at much higher risk of getting a cardiovascular event. And that especially when they are on statin, for example, medication. And I think that's something that in this medical field is of key importance, that we want to identify those that are on medication but still have high unmet clinical need. And that's what we can provide with this cellular readout. And I think it will be like more severe for those people that are older. But overall, it doesn't matter if this is like a young or an old person. If you have a poor cellular score, poor LDL uptake, then it's very, very likely that you need more effective treatment, for example, combination treatment, to reduce your risk. I mean, if you are FH, for example, and that's in a way a nice comparison. That's also what we think, like this is underlying. If you have a loss of function mutation in LDL receptor, you are homozygous for it. For example, you can develop a heart attack with below 20 years of age. So if you are heterozygous, you get a heart attack if you are, I don't know, in the range of 30 to 50 years. And we think that people that have no mutation in that and that still have a poor cellular score will have probably a similar outcome for that. And therefore, I think this is very like a unique opportunity, because all of these like more effective drugs are actually limited to people that already experienced a heart attack or already have a genetic diagnosis for FH, at least in Finland. If you want to prescribe the PCSK9 inhibitor therapy, you need to have a genetic test for FH, and you need to show that the doctor doesn't bring down blood cholesterol with all of the other existing medication. So, but then for primary prevention, there is no like reimbursement strategy for that. This medication can't get to people in primary prevention, even though there would be patients who would actually need it. So I think, I think that's a great opportunity actually to really bring precision medicine to these people, roll it out at large scale, and then get effective treatment to hopefully to everybody.
Thank you. That's indeed an important point. Coming back still to the legislation, in particular, we do have the GDPR, the General Data Protection Regulation for the European countries in respect to the biobanking law. What we heard that some of the countries do have, others do not. In Latvia, we have been trying to, and Vita is really the key person behind this, have been trying really to have the legislation accepted, and Finnish legislation was used as the model for this. How it is in United Arab Emirates? Do you also have specific laws for biobanking, for the data protection, or how it's embedded to the legislation?
Of Ministry of Health and Prevention, we have to follow the law, their law, according to the government law.
Thank you. That's very interesting, and I think actually the requirements are pretty the same. It's no huge difference.
Even when we are going to do something, we have to raise. Even if we're going to do anything, we have to raise a specific application to say want to do this. If they agreed, fine. If they don't agree, they say no. You have to correct this. You have to do that. You have to do this.
But do you need to go back with each of the questions to the individual and to ask for their acceptance?
Yes.
If you want to do another blood test, another sample, another study, each time you have to go back to the
even if we want to do, for example, let's say we're going to take 10 ml of blood and instead 8 ml. We have to get their approval. We cannot do that by our own.
Yeah, sure.
Because the editor, when they come, they could stop the program if there is any violation for that.
Mm-hmm. Okay, thank you. Any other questions? If no other questions, let's thank all the speakers. And the message from the organizers that everybody is welcome in the lobby to have a welcome drink. So it is still not the time we have to leave, but we can really meet and network and continue this.
Prof, we would like to thank you personally for your time and your effort, and distinguished attendees, and thank you for listening.
Absolutely. I'm joining also to the thanks to all the audience. And indeed, I think this has been a very exciting topic. And this year, it has been much better attended than the last year. So you are welcome to come back within a year from now, when I expect we will have even more participants.
Special thanks to Her Excellency Hanan Laleli, the Ambassador of United Arab Emirates in Latvia, and Mr. Khalid Zaabi. He is the Director of Government and International Relation. And I would like to thank all my colleagues who supported, who work in the Emirati Genome Program and M42. Thank you.
Thank you.